Welcome back to Bookfulness, and another deep dive into the foundational texts of evolutionary science. This is episode 10. We've just wrapped up Unit 2, which really dug into the core mechanics, how selection, drift, gene flow actually work, the engine room, so to speak.
And now we're making quite a pivot. We're heading straight into Unit 3, which looks at the products of evolution. So we're shifting from, let's say, the physics of the process to the actual results, the often spectacular things evolution has built.
And what a starting point. We're tackling one of nature's most fundamental, perplexing, and maybe most fascinating topics.
Sex.
Absolutely. It's everywhere, but when you really look at it, it's also incredibly complex and, frankly, costly.
Right.
Yeah.
It often seems, on the surface anyway, like a really inefficient way to pass on your genes, yet it dominates the living world. So our mission today, I think, is threefold. First, let's get a precise biological definition. What actually is a male versus a female? Second, we need to understand the sheer power of sexual selection, how it shapes organisms.
Right, the driving force behind some incredible traits. And finally, the big one. Tackle the core evolutionary puzzle. Why does sex even persist when it seems to have these huge built-in costs?
It's a journey that takes us from, microscopic gene swapping in bacteria all the way up to massive mammals locking horns. It forces you to think differently. Oh, and just a quick note before we dive in for you, the listener, we'll be mentioning quite a few figures from the text, like figure 10.1 or figure 10.21.
Yeah, these are really helpful for visualizing things.
They are, and you can find all these diagrams linked in the supplementary file. It's right there in the description for this deep dive.
Perfect. Okay, let's get into it. Part 1, defining sex, females, males, and diversity. The first thing the source material really emphasizes is that sexual reproduction isn't just one thing. It's this huge spectrum across life.
That's right. The core evolutionary function, the whole point, really, is mixing genes. Genetic recombination. And life has found, well, lots of ways to do that. Even organisms we don't typically think of as having sex, like bacteria or viruses, they do something called parisexuality.
Parisexuality. So it's not quite sex as we usually picture it.
Not in the sense of meiosis and distinct male-female gametes, no. But they achieve the same outcome. Genetic mixing. Bacteria, for instance, can do conjugation. They form a little bridge, pass DNA across. Or they can just absorb bits of DNA floating around from dead cells.
Just picking up genetic material from the environment.
Yeah, naked DNA. And viruses, well, if multiple virus particles infect the same host cell, their offspring are going to be genetic mashups of the parents. The key result is recombination.
So the fundamental goal, mixing genes, is achieved in these different ways.
Exactly. And then when you get to multicellular life, the structural diversity just explodes. It's not always straightforward male and female.
Right. Figure 10.1 shows some pretty surprising examples.
It really does. You've got hermaphrodites, organisms with both male and female reproductive organs. Think of those colorful sea slugs. Hypsilodoris bulky. They can kind of switch roles or even use both at once.
And then they're the ones that change sex completely during their lifetime. That seems, well, pretty alien to us mammals.
That's sequential hermaphroditism. The classic example is the clownfish. Amphiprion, like Nemo. They live in these strict social hierarchies. The biggest fish is the female. The next biggest is the breeding male. Everyone else is basically an immature male.
So what happens if the female dies?
The breeding male changes sex, becomes the new dominant female, and the next largest immature male quickly develops into the new breeding male. Sex is determined by social rank and size, not fixed chromosomes. It's fluid.
Wow. Okay, so with all this diversity, hermaphrodites, sex changers, bacteria swapping genes, what's the single unifying biological definition that separates male from female when you do have separate sexes?
It all boils down to one thing: gamete size, cell size. It's a concept called anisogamy.
Anisogamy.
It just means having two types of gametes, sex cells, that are distinctly different in size and form. It's fundamental. The sex that produces lots of small, usually mobile gametes, think sperm in animals, or pollen in plants, that's defined as the male. Quantity and motility are key.
Got it. Small and many.
And the sex that produces fewer, much larger, nutrient-packed gametes, eggs in animals, ovules in plants, that's the female. Investment and resources are the focus.
So it's purely about the size and number of the sex cells produced.
That's the core biological definition, yes.
But why? Why did evolution settle on these two extremes? Why not just, medium-sized gametes for everyone? It's called isogamy, right?
Exactly, isogamy. And it probably did exist early on. But it's thought to be an unstable state. Imagine everyone producing medium gametes. Now, what if a mutation pops up that lets an individual make slightly smaller gametes, but twice as many for the same energy cost?
Okay.
If those smaller gametes can still fuse successfully with a normal medium one, the resulting zygote is only slightly smaller, but the producer of the small gametes has doubled their potential offspring.
Ah, so there's an immediate advantage to making smaller, more numerous gametes. Selection favors that.
Yes, but at the same time, think about the other gametes. Once these tiny gametes are common, if a medium gamete fuses with one, the resulting zygote might not have enough resources to develop properly. So now there's selective pressure on the other gametes to become larger and more nutrient-rich to compensate.
Ah, okay. So one lineage gets smaller and more numerous, the other gets larger and more resource-heavy.
Precisely. It's called disruptive selection. It pushes the system towards two distinct strategies, two extremes: tiny, mobile sperm and large, resource-laden eggs. That difference, anisogamy, becomes the stable outcome.
That's a really neat explanation for such a fundamental split. So anisogamy defines the sexes based on gamete size, but how an individual becomes male or female, the determination mechanism, that varies a lot too.
Hugely. Figure 10.3 gives a glimpse. We're familiar with chromosomal systems like XXXY in humans or ZZZW in birds, but there are others. Some insects, like bees and ants, use ploidy females developed from fertilized eggs, males from unfertilized ones, and then there's temperature.
Right, the temperature-dependent sex determination in reptiles.
Yeah, in many turtles, crocodilians, some lizards, the temperature during a critical incubation period determines the sex. Sometimes cooler temps make males, warmer makes females. Sometimes it's the reverse, or extremes make one sex and intermediate temps make the other.
So the environment itself throws the switch, maybe ensuring a mix of sexes across different nesting sites.
That's a likely hypothesis, yeah. And the physical differences that result from being male or female, the sexually dimorphic traits, they can range from almost nothing
to the completely bizarre. Which brings us back to that anglerfish example in figure 10.4.
Ah, the deep-sea anglerfish. It's just the ultimate example of how extreme selection can get. Remember, the male is defined by making tiny sperm, the female makes the big eggs, and the deep sea is vast, dark. Finding a mate is incredibly difficult.
So the pressure to hold on to a mate once found is immense.
Astronomical. So when a tiny male, and they can be minuscule, like less than 1% of the female size finds a female, he doesn't just mate and leave, he bites on. Their tissues fuse, their circulatory systems merge, his eyes, jaws, internal organs, they all degenerate, except his tests. He becomes essentially a parasitic sperm factory attached to her body.
He literally gives up his entire independent existence just to reproduce.
Survival becomes secondary to reproduction. The female provides life support and a guaranteed mating opportunity. In that environment, it's the strategy that won out. His whole body plan is shaped by anisogamy and mate scarcity.
Wow. The fact that selection favors him becoming just an appendage really drives home the power of reproductive pressure, which leads us perfectly into part 2. The power of sexual selection, the force behind these often extreme traits.
Right. Once you have anisogamy, the difference in investment, you automatically get competition. And that competition creates its own selective force distinct from just surviving day to day.
Let's clarify the traits again. There are primary sexual traits, the gonads, the basic plumbing needed for reproduction.
Your testes, ovaries, genitals.
And then secondary sexual traits.
Yeah.
That's pretty much everything else that differs between the sexes, right? The peacock's tail, the deer's antlers, the bird's song.
Exactly. And it was Darwin who spotted that these secondary traits often seem bad for survival. Big antlers make it harder to escape wolves. Bright feathers attract predators. He proposed sexual selection as the explanation. Selection arising from differences in mating success among individuals of the same sex.
So it's not about surviving better, it's about mating more.
Precisely. Darwin noted these traits are often exaggerated, usually in males, often appear only during the breeding season, and are absent in youngsters. The key insight is that sexual selection can favor traits that actually reduce survival if the boost in mating success is big enough to outweigh the cost.
It's a trade-off, a reproductive gamble.
Totally. And the classic experiment that really nailed this down came much later, with the long-tailed widowbird Euplectes prone. It's shown in figure 10.5, a beautiful piece of work.
This one's really fundamental for understanding how we test these ideas, isn't it? Walk us through how they did it.
Okay, so male widowbirds, they're striking blackbirds, and the males have these incredibly long tail feathers, sometimes over 1/2 meter. The hypothesis was females prefer longer tails, and this drives the evolution of the trait despite the cost. Researchers went out onto the African savanna, caught a bunch of territorial males, and divided them into 4 groups. They made sure the groups were matched for territory quality and the male's initial condition.
Smart. What were the 4 treatments?
First, one group had their tails cut short, really short. Second, another group had their tails artificially lengthened. They actually glued on the feather tips cut from the first group, making the tails even longer than normal.
Okay, short and super long.
Third, a control group. Caught, measured, released, tail untouched. And fourth, crucially, a sham control. These birds were caught, handled, had their tails cut, and then immediately glued back together to the original length. This controlled for the stress of being handled and having glue applied.
Right, so isolating the effect of tail length itself and the results.
Crystal clear. They monitored the territories and counted how many active nests each male attracted. The males with the artificially lengthened tails attracted significantly more females than any other group, and the poor guys with the shortened tails, they attracted the fewest.
So longer tails, even artificially longer, meant more mates. Independent of the male's actual quality, just the signal itself worked.
Exactly. It showed that female preference for this trait, which is almost certainly a burden for flight and energy, is strong enough to drive its evolution via sexual selection. The reproductive benefit outweighs the survival cost.
This highlights that constant mating trade-off. There's always this tension between maximizing mating and maximizing survival. What's another good example? Maybe using sound.
The túngara frog, Physalimys pustulosus, is a great case, shown in figure 10.6. Male frogs call to attract females. The basic call is a simple whine. It works. But often, males add a more complex, low-frequency sound at the end, a chuck, so it goes whine chuck. And females really prefer the complex call.
Okay, so the chuck is like the long tail, more attractive. What's the downside?
The downside is deadly. The chuck sound is much easier for predators to locate, specifically the fringe-lipped bat, Trichops cirrhosis, which specializes in eating frogs. Adding the chuck makes the male frog basically scream, "Eat me!" to the bats.
So the poor frog is caught in a bind.
Totally. Chuck and risk getting eaten right now, but maybe get a mate. Don't chuck, stay safer, but probably lose the female to a risk-taking neighbor. The male is constantly weighing that risk versus reward.
Which brings up a point. If the chuck is so risky, why hasn't female preference evolved to ignore it or even dislike it? Wouldn't that favor males who survive longer?
That's a fantastic question, and it points to deeper reasons for the preference. Maybe the ability to produce a good chuck, despite the risk, is an honest signal of the male's quality of his health, his vigor. So females choosing the chuck are getting good genes for their offspring. Or maybe the bats are a relatively recent pressure. But whatever the reason, there's this tension. Natural selection, predation, pushing for simpler calls. Sexual selection, female preference, pushing for complex calls. The call length we observe is likely the equilibrium point.
This pressure, this competition and elaborate display, it usually seems more intense on males. Why is that typically the case?
It comes down to 2 key concepts, Bateman's principle and the operational sex ratio, OSR. A.J. Bateman's work back in the 40s with fruit flies suggested a general pattern. A male's reproductive success tends to increase directly with the number of females he mates with.
Right, because sperm is cheap to produce, energetically speaking. He can potentially father many offspring.
Exactly, whereas a female's success is typically limited by the number of eggs she can produce or the number of offspring she can raise. Making eggs is costly. Gestation, parental care, these are huge investments. Mating with more males beyond what's needed for fertilization often doesn't increase her total offspring number significantly.
So males gain more fitness from multiple matings than females do.
Generally, yes. This difference in potential reproductive rates means that selection for traits that increase mating success, sexual selection, usually acts more strongly on males. They have more to gain.
And the operational sex ratio, OSR, adds to this.
It magnifies it. The OSR isn't just the total number of males and females, it's the ratio of males who are ready to mate right now to females who are receptive right now. And because females often have long periods where they're unavailable developing eggs, pregnant, caring for young, the pool of receptive females at any given moment is usually much smaller than the pool of sexually active males.
Making receptive females a scarce resource.
Precisely. So if you have lots of males competing for very few available females, the competition becomes incredibly intense. That's what drives the evolution of male weapons, elaborate displays, and risky behaviors.
And the exceptions really prove the rule here, don't they? When the roles in parental investment are reversed.
Absolutely. Look at species where the male makes the larger investment, figure 10.8. Suddenly, he becomes the limiting resource, and the females compete for access to him. Take red phalaropes, a type of shorebird. The females are larger, more brightly colored, and they fight aggressively over the duller males.
Why the switch?
Because the male phalarope does all the incubation and chick rearing, his time and energy are limited, so the female's success depends on how many males she can persuade to raise her clutches. She competes, he chooses.
Wow. And seahorses, too.
Male Australian seahorses are famous for it. The female lays her eggs into a pouch on the male's belly, and he carries them, provides nutrients, and gives birth. He's the pregnant one. So males are the scarce resource. Females compete, performing elaborate courtship displays to convince a male to accept their eggs. It perfectly illustrates that competition follows investment, not biological sex per se.
That's really clear. Okay, let's dig into the specific ways males compete. Part 3. Male-male competition, interference. This is the first major mode of sexual selection.
Right. This is where males directly interfere with each other's attempts to mate. It often involves physical contests, leading to selection for size, strength, and weaponry.
The classic image is probably male combat, like those red deer stags battling during the rut, right? Figure 10.9a shows this. What exactly are they fighting for?
For red deer, Cervus elaphus, it's all about controlling access to females. During the breeding season, or rut, dominant males try to gather and defend groups of females called harems. The fighting is intense, often ritualized, but injuries are common and can be severe blindness, broken antlers, even death.
Seems incredibly risky. Why is the payoff worth it?
Because reproductive success is massively skewed. The top stags, the winners of these fights, might sire a huge proportion of the offspring born the next year, maybe 50% or more, from just the top 10% of males. Meanwhile, many males, maybe nearly half, might fail to reproduce at all in a given season. The potential reward for winning is so enormous, it justifies the risk and the cost of growing those huge antlers.
So selection favors being a good fighter, even if it shortens your overall life. And it's not always directly over females. Sometimes it's about resources females need.
Exactly. That's the ecological factor insight. How resources are distributed matters hugely. Look at stag beetles, Lucana cervus and figure 10.9b. Those males have enormous mandible antlers. They use them to fight over specific sites like rotting logs or tree sap flows that females need for feeding or laying eggs.
So controlling the resource means controlling access to females.
If the essential resource is clumped together and defensible, like a good log, then selection will favor males who can monopolize it. If the resource was spread out evenly, that kind of intense fighting wouldn't pay off as much.
Okay, so direct combat is one strategy. But the competition doesn't necessarily stop once mating has happened, does it?
Not at all. Welcome to the world of post-copulatory competition, most notably sperm competition. If females mate with multiple males, which is very common, then the competition continues inside the female's reproductive tract, whose sperm actually fertilizes the egg.
And males have evolved some pretty amazing, sometimes quite gruesome ways to win this internal battle. The damselfly example in figure 10.11 is striking.
Oh, it's fantastic. The male damselfly's penis isn't just for delivering sperm. It has evolved these specialized hooks, spines, and scoops. Before he transfers his own sperm, he uses this modified structure to literally scrape out or displace the sperm left by any previous males the female might have mated with.
Physically removing the competition.
It's quite mechanical. By clearing the storage organ, he dramatically increases his own chances of paternity. Other strategies include toxic seminal fluids that kill rival sperm, or leaving a physical barrier called a copulatory plug.
It's an internal arms race.
It absolutely is. And you see parallels in plants too. Pollen competition. When pollen lands on a flower's stigma, multiple grains might start growing pollen tubes down towards the ovules. It's a race. The fastest pollen tube achieves fertilization.
Okay, but what if a male just isn't big or strong enough to win fights or defend resources? Surely evolution provides other options.
Definitely. Direct confrontation is costly and risky. If you're likely to lose, selection favors finding another way. This leads to alternative mating strategies. Often you see smaller or less dominant males using tactics like sneaking or mimicry. The marine isopod, Paracerce sculpta, living in sponges, is a brilliant example. Figure 10.10 shows this. There are 3 distinct male forms.
3 types of males in one species.
Yep. You have the large alpha males. They're big, aggressive, and they guard harems of females inside the sponge cavities. Standard fighters. Then you have beta males. These guys are medium-sized, and they look almost exactly like females. They use this mimicry to trick the alpha into letting them into the sponge.
Female mimics. Sneaky. And the third type?
The tiny gamma males. They're so small they can just slip past the alpha's defenses unnoticed. Stealth is their game.
So you have fighting, mimicry, and stealth all coexisting. Why doesn't one strategy just win out?
It's likely frequency-dependent selection. The success of each strategy depends on how common the others are. If mimics, betas, become too common, alphas might get better at spotting them, reducing the mimic's success. If fighters, owls are everywhere, competition is fierce, and maybe being a tiny sneaker, gamma becomes the best option. Each strategy does best when it's relatively rare, which maintains the diversity.
That makes sense. A balanced portfolio of strategies. Now, there's one final, really dark aspect of male competition mentioned. Infanticide. Figure 10.12 touches on this.
Yes, infanticide, the killing of dependent offspring by males, is perhaps the most extreme and disturbing manifestation of sexual selection.
It's often associated with mammals like lions.
But lions are the classic, most studied example. When a new male, or a coalition of new males, takes over a pride by ousting the previous resident males, they very often kill any young, unweaned cubs fathered by their predecessors.
It seems incredibly brutal. Is there an evolutionary logic behind it?
There is, unfortunately. It's not random violence, it's a reproductive strategy. A female lion won't become receptive to mating again while she's still nursing cubs. The lactation hormones suppress her cycle. By killing the cubs, the new males remove that hormonal break. The females come back into estrus, become sexually receptive much sooner, sometimes weeks or months earlier than they would have otherwise.
So the new males can start siring their own offspring faster.
Exactly. Male tenure in a pride can be short. They need to maximize their own reproductive output during their window of dominance. It's a stark example of selection favoring traits that increase a male's gene transmission, even at the horrific cost of existing offspring.
A chilling reminder that evolution acts on reproductive success, sometimes with outcomes we find morally abhorrent. Okay, let's shift perspective completely now, away from male conflict and towards the other side of the equation. Part 4. Female choice, preference. The second main driver of sexual selection.
Right. If male competition is about fighting off rivals, female choice is about females actively selecting mates based on certain characteristics. Their preferences act as a powerful evolutionary filter.
And these preferences aren't just random whims, are they? The source material emphasizes that the preferences themselves evolve. How do we know this?
There's good evidence. First, preferences can be heritable. Just like a male trait, like tail length, can be passed down. A female's tendency to prefer that trait can also have a genetic basis, meaning selection can act on it. Second, we see patterns like reinforcement and speciation. Females of one species often evolve strong preferences for males of their own species and actively reject males of closely related species. This shows preferences evolving as isolating mechanisms.
Okay, so preferences evolve. The book lays out 4 main reasons why a particular preference might evolve. Let's start with simplest. Mechanism 1, direct benefits.
This is the most straightforward. Females choose males who provide them with some immediate, tangible resource that directly increases her own survival or the number or quality of offspring she can produce.
Like a gift.
Exactly. The classic example is a nuptial gift. In many insects, for instance, the male provides a spermatophore, a package containing sperm, yes, but often also packed with proteins and nutrients. The female eats the spermatophore casing, gaining valuable resources she can use for egg production or her own maintenance. Choosing a male with a bigger gift directly benefits her fitness.
So the preference gene spreads because it directly helps the female herself. Makes sense.
Here, selection on the preference allele is direct.
Okay, now mechanism 2. Pleiotropic effects, sometimes called perceptual or sensory biases. This one feels a bit different. The preference exists before the trait.
It's a really cool idea. The argument is that a female's preference for a certain male trait might have evolved as an accidental side effect, a pleiotropic effect of her sensory system being tuned for something else entirely, like finding food of a certain color or avoiding predators with specific patterns.
So her brain is already wired to notice, say, the color orange because orange fruits are nutritious. And then if a male happens to evolve an orange spot,
she's already predisposed to notice it and perhaps be attracted to it. The preference came first as part of her adaptation for foraging or survival. The male trait then evolves to exploit this pre-existing bias.
Is there evidence for this?
Yes, the fish example is key here. Swordtail fish, Xiphophorus, males have that long sword extension on their tail and females prefer it. Researchers looked at a closely related genus, Priapella, where males don't have swords. They experimentally attached artificial swords to male Priapella, and guess what? The female Priapella, who had no evolutionary history with swords, showed a strong preference for the males with the artificial swords.
Wow, the bias was just sitting there latent.
Exactly. The preference allele was likely selected for its role in the non-mating sensory context, making selection on the preference direct in that sense, but the male trait evolves later to tap into it.
Fascinating. Okay, now we move to mechanisms involving indirect selection. These seem a bit more complex. Let's start with mechanism 3, good genes or indicator traits.
Right. Here, the idea is that females choose males based on traits that reliably signal the male's underlying genetic quality. She's not choosing for herself directly, but for the genes her offspring will inherit. Genes for things like disease resistance, foraging efficiency, overall health, and vigor.
But how can she tell? How does a trait become an honest indicator of good genes?
The key is that the indicator trait must be costly to produce or maintain. Think of the peacock's tail again. Glowing and displaying that enormous, vibrant train takes a huge amount of energy and resources. Only a male who is genuinely healthy, well-fed, and parasite-free can afford to produce such a magnificent display. A sick or weak male just can't manage it. The costliness ensures honesty. The signal can be trusted.
Okay, so the flashy trait indicates good underlying genes. How does the female preference for that trait then evolve? This is where indirect selection and this term linkage disequilibrium come in, right?
Exactly, and this is crucial but can be tricky. Indirect selection means the preference gene itself doesn't necessarily provide a direct fitness benefit, but it increases in frequency because it gets associated with other genes that do. Linkage disequilibrium, LD, just means that alleles at different gene locations are non-randomly associated. They tend to be inherited together more often than you'd expect by chance, maybe because they're physically close on a chromosome or just because of selection patterns.
So walk me through it. Let's say P is the allele for preferring long tails, and G is an allele for good parasite resistance that helps produce long tails.
Okay. If females with the P allele choose males who, because they have the G allele, manage to grow long tails, then their offspring are likely to inherit both the P allele from mom and the G allele from dad.
So the preference gene P and the good gene G end up linked in the offspring. They're in LD.
Precisely. Now, natural selection strongly favors the G allele because it confers parasite resistance and thus higher survival reproduction. Since the P allele is statistically traveling with the G allele, P gets pulled along for the ride. The frequency of the preference allele P increases, not because P itself does anything useful, but because it's correlated with the beneficial G allele. That's indirect selection on the preference.
That's much clearer. The preference gene hitches a ride with the good genes it helps select. It's the correlation that matters. Okay, final mechanism, and perhaps the most dramatic. Mechanism 4, Fisher's runaway sexual selection.
This is Ari Fisher's famous idea. Like the good genes model, it starts with a genetic correlation linkage disequilibrium between a male trait, T, say tail length, and the female preference for it, P. Imagine some females develop a slight preference for slightly longer tails. T. Males with longer tails get chosen more often, so they have more offspring.
Okay, similar start. But here, the trait doesn't have to indicate good genes.
Correct. It could just be arbitrary initially. The key is the feedback loop. Sons inherit the genes for the longer tail, T, from their successful fathers. Daughters inherit the genes for the stronger preference, P, from their mothers. Now, females with the stronger preference, P, have sons with the longer tails, T, who are then preferred by the next generation of females carrying allele P. This genetic correlation fuels itself.
It becomes a self-reinforcing cycle.
Exactly. The preference drives the evolution of the trait, and the evolution of the trait reinforces selection for the preference. It runs away on its own momentum. Selection favors increasingly extreme traits and increasingly strong preferences purely because of this genetic coupling.
So why don't tails just get infinitely long? What stops the runaway?
Natural selection eventually puts the brakes on. At some point, the trait becomes so exaggerated, like a ridiculously long tail, that the survival costs, difficulty flying, attracting predators, energy drain become too high. The runaway process starts when the disadvantage in terms of survival perfectly balances the advantage in terms of mating success. It explains how selection can produce these incredibly elaborate, seemingly non-adaptive ornaments.
And just to recap the key difference, direct benefits in sensory bias involve direct selection on the preference allele itself because it provides an immediate benefit or exploits an existing sensory function. Good genes in Fisher's runaway involve indirect selection on the preference because it spreads due to its genetic correlation with other selected genes, either good survival genes or the attractive trait itself.
That's the critical distinction, yes.
Fantastic. This really shows the complex ways female choice shapes evolution. Okay, let's shift again. Part 5. Sex ratios and the enigma of sex.
First, let's tackle that remarkably consistent 1.1 sex ratio we see in most species. From a purely population growth view, it seems weird, right?
Yeah, you'd think populations would grow fastest if they produced mostly females and just enough males to get the job done. Maybe like 10 females for every male.
Logically, yes, since only females actually produce offspring in most cases. But nature almost always settles on a 50-50 split, at least at conception or birth. Why?
This was explained by Fisher, wasn't it? Using frequency dependence.
Yes, R.A. Fisher's argument is incredibly elegant, shown conceptually in figure 10.16. It relies on one simple fact. Every offspring has exactly one mother and one father. This means the total reproductive success summed across all males in a generation must equal the total reproductive success summed across all females.
Okay, so what happens if the ratio becomes unbalanced, say way more females than males?
Imagine a population that's 80% female, 20% male. Because every resulting offspring still needs one father, those few males are, on average, contributing to far more offspring than the average female is. Each male potentially fathers offspring from multiple females. In this scenario, the average reproductive success of a male is much higher than that of a female.
Ah, so being the rare sex gives you a huge advantage.
Exactly. Now, consider a parent carrying a mutation that causes them to produce more offspring of the rare sex, in this case, sons. Their offspring will, on average, have much higher reproductive success than the offspring of parents producing mostly females. That mutation will spread like wildfire through the population, producing more males.
Until the ratio gets back to 1.1.
Precisely. Once it's 1.1, the average reproductive success of males and females is equal, and the selective pressure disappears. It's a powerful self-correcting mechanism based on frequency-dependent selection.
So stable. But are there exceptions? Can parents ever adjust the ratio?
There are exceptions, often linked to specific environmental conditions where one sex might provide more fitness benefits than the other. Figure 10.18 shows examples like fig wasps, but the Seychelles warbler is a great vertebrate case.
What do the warblers do?
Acrocephalus secalensis. These birds defend territories, and territory quality varies. On high-quality territories with lots of food, pairs predominantly produce daughters, sometimes close to 90% daughters. Why? Because daughters often stay on the parental territory as helpers, assisting in raising future siblings. This is beneficial if there are enough resources.
But on poor territories.
On poor territories, helpers would just be extra competition for scarce food. So pairs on low-quality territories produce mostly sons. Sons disperse away from the poor territory to find their own opportunities. The parents adaptively adjust the sex ratio to maximize their offspring success given the local conditions.
Clever. Okay, so the 1.1 ratio is generally stable due to frequency dependence, but adaptable. This stability, though, just makes the fundamental question sharper. The evolutionary enigma of sex. Why bother with males at all?
This is the big one. Given the cost, why is sexual reproduction the norm? The main alternative is asexual reproduction, like parthenogenesis, where females produce offspring from unfertilized eggs, basically cloning themselves. Figure 10.12 mentions this.
And asexual reproduction does occur, but it seems rare and often short-lived evolutionarily.
It pops up in about 1% of plant species and maybe 0.1% of animals. Think whiptail lizards, some fish, dandelions. But most asexual lineages seem to be evolutionary twigs. They don't persist for long geological timescales. The big exception everyone points to are the beetaloid rotifers, tiny aquatic creatures. Fossil and genetic evidence suggests they might have been entirely asexual for tens, maybe over 100 million years. How they manage it is still a bit of a mystery.
But for most, asexuality seems like a losing long-term strategy. Why? Especially given the huge advantage it seems to have the famous twofold cost of males. We really need to hammer this home. Figure 10.21.
Absolutely. The twofold cost is the central paradox. Let's break it down. Imagine an ancestral population where some females reproduce sexually and some mutation allows others to reproduce asexually. Cost number one, the cost of meiosis, or cost of genome dilution. A sexual female passes only 50% of her genes to each offspring. An asexual female passes 100%. Cost number two, the cost of producing males. In a sexual population, roughly half the offspring are males, who themselves cannot produce offspring. They only contribute genes. Asexual populations consist entirely of reproductive females.
So the asexual lineage grows.
Twice as fast.
Yeah.
Generation after generation, the asexual females churn out twice as many reproductive individuals as the sexual females. Any asexual mutant should rapidly outcompete the sexual individuals and take over the population in relatively few generations.
So why hasn't this happened everywhere? Sex must provide some massive benefit to overcome this immediate huge disadvantage.
Exactly, and that benefit must be linked to the outcome of sex. Genetic recombination, the shuffling of genes.
Is there evidence that mixing genes is more beneficial in certain environments?
Yes, organisms that can switch between modes provide clues, like the water flea, Daphnia. In stable summer pond conditions, they reproduce asexually like crazy, maximizing population growth. But come autumn, when conditions become unpredictable, or before winter dormancy, they switch to sexual reproduction. They produce genetically diverse resting eggs that can survive the harsh conditions. This suggests sex is favored when the environment is changing or challenging.
Okay, changing environments favor variation. But simply shuffling genes can break up good combinations too, right? Like shuffling a winning hand of cards might give you a worse hand. Why is the mixing itself so valuable?
This leads us beyond just coping with physical changes and towards the dominant theory, the Red Queen hypothesis.
Named after the character in Through the Looking Glass.
That's the one. The Red Queen tells Alice it takes all the running you can do to keep in the same place. In evolution, this means species are constantly locked in arms races with their enemies. You have to keep evolving just to maintain your current level of fitness relative to antagonists.
And the main antagonists driving the need for sex are
Parasites and pathogens. Viruses, bacteria, fungi, parasitic worms. These things typically have much shorter generation times than their hosts, so they can evolve incredibly quickly to overcome the host's defenses.
So if a host reproduces asexually, it produces clones.
Exactly. And if one clone becomes common because it's successful, it becomes a huge static target for parasites. The parasites quickly evolve ways to exploit that specific common genotype. Sex, through recombination, constantly shuffles the host's genes, creating new, rare combinations of defense alleles in every generation. It's like constantly changing the locks so the rapidly evolving parasite keys no longer work.
So sex is crucial for staying one step ahead in this coevolutionary arms race.
That's the idea. It generates the necessary genetic novelty to fight off rapidly evolving enemies.
And the evidence. The New Zealand mud snail study, figure 10.22, is often cited as strong support, isn't it?
It's considered a classic test, yes. Potamopyrgus antipodarum. These snails live in lakes and streams, and crucially, populations contain both sexually reproducing snails and various asexual clonal lineages, all living side by side. Researchers looked at snail populations facing different levels of infection by sterilizing trematode worms, a type of parasite.
And what did they find?
A strong correlation. In habitats where parasite infection rates were high, the proportion of sexual snails was also significantly higher. Where parasites were rare, asexual clones dominated. Even more compellingly, they found that in those high infection areas, sexual females often had lower infection rates than the common asexual clones. And in some cases, the measured fitness advantage of being sexual, due to lower parasites, was roughly twofold enough to exactly offset the cost of producing males.
Wow. So the benefit of recombination in fighting parasites seems quantitatively sufficient to explain the maintenance of sex.
It's powerful evidence supporting the Red Queen as a major driver. Sex is maintained, at least in part, as an anti-parasite strategy.
That really clarifies the enigma. Okay, let's wrap up with part 6. Why sex prevails, focusing on the specific genetic advantages that recombination provides beyond just generating novelty.
Right, sex isn't just about creating new combinations, it's also about breaking up bad ones and allowing beneficial mutations to spread more efficiently. It helps reduce selective interference.
What does that mean, selective interference?
In asexual lineages, genes are permanently linked together on the chromosome. This can cause problems. One issue is clonal interference. Imagine two different beneficial mutations arise independently in two different asexual clones. Because there's no recombination, these two mutations can't be brought together in the same lineage. The two clones end up competing against each other.
And the best mutation might lose out.
It might. If the clone carrying the slightly better mutation happens to also carry other slightly disadvantageous genes, a worse genetic background, it might be outcompeted by the clone with the slightly less beneficial mutation but a better overall background. Sex breaks this linkage, allowing beneficial mutations from different lineages to combine.
And there's a related idea, the Ruby and the rubbish effect. Figure 10.24 illustrates this.
It's a great visual metaphor. Imagine a really valuable, beneficial mutation, the ruby, arises. But in an asexual lineage, it happens to arise in an individual that already carries a load of slightly harmful mutations, the rubbish. Because the ruby is permanently stuck with the rubbish, natural selection might eliminate the whole lineage if the combined fitness effect is negative. The valuable ruby gets thrown out with the trash.
How does sex fix this?
Recombination. Sex allows that ruby mutation to be separated from its original rubbish background and combined with a cleaner, fitter genetic background. This allows selection to preserve the beneficial mutation much more effectively.
Is there experimental proof of this?
Yeah.
The yeast study?
The yeast experiments, figure 10.25, are pretty convincing. Researchers let replicate populations of yeast adapt to a lab environment. Some were forced to reproduce asexually, others could reproduce sexually via recombination. They found that while both adapted, the asexual populations accumulated deleterious mutations alongside the beneficial ones. The beneficial mutations essentially dragged the rubbish along with them because they couldn't be uncoupled.
And the sexual populations.
The sexual populations, thanks to recombination, were able to purge these deleterious mutations more effectively while still fixing the beneficial ones. They adapted more cleanly without accumulating as much genetic load. By the end, the asexual lines had fixed significantly more harmful mutations.
This accumulation of bad mutations in asexual lines relates to Muller's ratchet, right?
Yes, Muller's ratchet is the idea that in a finite asexual population, the class of individuals with the fewest deleterious mutations can be lost by chance, genetic drift. And because there's no recombination, that fittest class can never be regenerated. The only way is forward, accumulating more mutations. The ratchet clicks, increasing the mutational load over time. Sex, by allowing recombination, can recreate those low mutation genotypes, effectively stopping or reversing the ratchet.
So asexuality leads to inevitable genomic decay over long timescales. Is there a real-world example of this we can see?
Absolutely. Look no further than our own Y chromosome, figure 10.27. For most of its length, the Y chromosome doesn't recombine with the X chromosome. It's essentially passed down like an asexual lineage from father to son.
And what's happened to it over evolutionary time?
It has decayed dramatically. Compared to the X chromosome, the Y has lost the vast majority of its original genes. It's shrunk in size and accumulated a lot of non-functional repetitive DNA. It's a textbook example of what happens without recombination, Muller's ratchet, and the ruby in the rubbish effect in action over millions of years.
A pretty stark warning about the long-term costs of asexuality. Okay, one final genetic benefit of sex. Avoiding inbreeding.
Right. Sex inherently involves combining genomes from two parents, which usually promotes outcrossing. This helps avoid the negative consequences of inbreeding, which is mating between close relatives, or the extreme case in plants, selfing, which is self-fertilization.
And the main problem with inbreeding is inbreeding depression.
Yes. Most populations carry hidden deleterious recessive alleles. These alleles only cause harm when an individual inherits two copies, is homozygous. Mating with a close relative dramatically increases the chance that offspring will inherit two copies of the same deleterious recessive allele derived from a recent common ancestor. This exposes the harmful effects, leading to reduced fitness, lower survival, fertility, etc., which is inbreeding depression.
How bad can it be? The white campion example.
The study on white campion, Silene latifolia, figure 10.29, found severe effects. Offspring produced by mating close relatives, like siblings, had fitness reduced by as much as 80% compared to offspring from unrelated parents, outcrossing.
80%? That's a massive selective pressure against inbreeding.
Huge. And you see the effects in humans too, of course. For example, recessive genetic diseases like Tay-Sachs are much more common in offspring of first cousin marriages than in the general population, illustrating the real medical consequences of exposing these hidden alleles. Selection strongly favors mechanisms that promote outcrossing,
which sexual reproduction inherently facilitates. Okay, that brings us to the end of this deep dive. We've journeyed from the very definition of male and female based on gamete size anisogamy through the incredible power of sexual selection, both male competition and female choice, shaping everything from antlers to anglerfish.
And then tackled the big why. Why pay the twofold cost of males? We explored the Red Queen hypothesis, the arms race with parasites, and the fundamental genetic benefits of recombination escaping Muller's ratchet, combining beneficial mutations, purging deleterious ones, and avoiding inbreeding depression.
It really covers the full sweep of the evolution of sex. That wraps up Chapter 10. Next time, in episode 11, we'll continue in Unit 3 by looking at Chapter 11, focusing on life histories. We'll be asking, how does evolution shape the entire life cycle of an organism to maximize fitness?
Yeah, we'll get into things like trade-offs between reproduction and survival, why some species have lots of tiny offspring and others have few large ones. Think R versus K selection and concepts like optimal clutch size, how organisms allocate limited resources across their lifespan.
Sounds like a great follow-up. How evolution optimizes the business of living and reproducing. And now for our final provocative thought for you to consider. Linking back to today's discussion on the Red Queen, we learned that sex is maintained, in large part, as a defense against rapidly evolving parasites and pathogens, especially in changing or unpredictable biological environments. So think about this. Are modern humans, with our high population densities and unprecedented levels of global travel, essentially creating a superhighway for pathogens to spread and mix? Are we inadvertently accelerating the very evolutionary arms race that makes sexual reproduction so essential for our species' long-term health?
Are we turning up the speed on the Red Queen's treadmill ourselves? A fascinating, maybe slightly unnerving thought to end on.
Indeed. Food for thought. Thanks for tuning in to Bookfulness. Join us next time for episode 11.
See you then.